This meeting is being recorded.
Good evening, Miss. We'll start. Okay, just 2 minutes. Can you mute yourself? Actually, okay, thank you. Okay, welcome everyone. Welcome to this yet another session of Spectratix knowledge series. Many of you have joined us previously. I can see many familiar names here. Though I don't know anyone by the face because no one turns on their video, so I know them only by their names. It means most of you know me by now. Because this is the 15th or 16th edition that we are running this Spectratix knowledge series. As far as Spectratix knowledge series goes, it's a series of events or a series of sessions which we organize on every Saturday. Here we have experts from different fields related to pharmaceuticals and associated fields, including nutraceuticals, Ayurveda, homeopathy, everything. We try to give the audience a perspective as to what new things are happening in the field of research and also the industry, academically also what things are happening, so that everyone keeps themselves updated. It's just for 1 hour that we meet, and it has become, I always keep repeating this, that it has become a science club where the discussions are open, wherein you can always give your suggestions in terms of your topics. Whatever topics you want us to cover, we'll be very happy to cover. So today I'm really happy to host this event known as Unlocking Nature's Pharmacy Drug Discovery from Natural Sources. Beyond that, I am very happy that Dr. Tina Merlin ma'am has accepted our invitation. She is my own professor. Whatever knowledge I have, it's all thanks to the wonderful professors that I had throughout my academic career. So, introducing Dr. Tina Merlin, she's a very accomplished professor. She is currently working as assistant professor of biochemistry at Mark Athanasius College for Advanced Studies, Tiruvalla, that is MACFAST, which is an autonomous institute under MG University, Kerala, India. And she holds advanced academic qualifications in biochemistry and has over a decade of teaching experience and significant research experience also in her own field. Her research interest spans across phytochemistry, inflammation biology, natural products, pharmacology, medicinal plants, and computational in silico analysis. Her area is that she integrates traditional knowledge with modern scientific techniques. Whoever had the opportunity to learn from her knows these things, that she's a very wonderful professor. She has established works on topics such as anti-inflammatory effects on herbal decoction, antioxidant toxicity or activity, and the biological evaluation of your traditional formulations. Her scientific contributions have earned her many national recognitions, including Young Ethnopharmacology Award from the Society of Ethnopharmacology. And this honor is given because of her research excellence and impact in the field of natural product science and anthropharmacology. She has led several research projects in the form of a principal investigator. She has worked, she had worked on the Women Scientific Project, which was funded by Kerala State Council for Science, Technology and Environment. And she's continuously mentoring many students in their academic research activities and also many startup related activities. In both academic and research roles, she continues to advance understanding at the intersection of biochemistry, traditional medicinal knowledge, and modern analytical science. So we are fortunate that she accepted our invitation. I am again thankful to her. It's all over to you, ma'am. I will present. You just tell me.
Hello, Sujit, am I audible?
Clear, ma'am. Clear.
Yeah, clear. Okay. Thank you, Sujit. Just as you said that I am also very happy that I got a chance through my student to be a part of such a noble thing that your company is doing. So first of all, thanks to Spectratix and definitely my student Sujit, because we need to discuss such things. It's very important because we know how actually our life has turned, and it's important to live, I mean, very safely in this present world. So I chose this topic, that is unlocking nature's pharmacy. Now, see, long before all these modern laboratories all developed, before that, nature was busy doing some things. I mean, nature was trying to unlock some real what secrecy was happening. Some molecules were being made, and it was all happening in silence. And we people later tried to think that everything was discovered by us. Now, definitely, pharmaceutical companies have a role, but still, all this, I mean, whatever compounds that have been discovered now, first was curated by nature. And now, this discovery of nature has to be taken into consideration. So, in today's talk, I'm having a bad throat, sorry for it. So, in today's talk, I will discuss how natural products continue to shape this modern discovery, how technology is transforming this field, and why countries like India have to take the first opportunity and unique opportunity to lead the future breakthroughs. Sujit, can we go to the next slide? Yeah, next step, please. So we actually know that our 65% of the global population is actually depending on natural products. Now that is a study by WHO. Now, why natural products? That is one important thing. That why discovery matters today. Now these natural compounds, they have a very important one. My own PhD work was on natural products. Now this demand of the compound discovery from natural products is very important, definitely for maintaining the well-being, the health of humans. Now, and why for? Because we know we are facing a global challenge, right from antibiotic resistance to rising as we personally we know that one or the other person, we just hear that some person is I mean suffering from cancer, or there is some metabolic, we just wonder. We found that person healthy just 2 days back, and then we come to know that that person is now suffering from some kind of cancer. So it is becoming that very, I mean, it's rising like anything. So drug discovery is going on, important. It matters today. So this limitation to why this drug discovery is not happening is definitely we have to find an effective solution that can be brought up to the market and can be utilized by everyone. Everyone in terms of, I mean, whatever drugs are present in the market, some definitely they cause lot amount of side effects. Now those side effects may make us think that should we use it or not. So moving on to drug discovery will definitely help us to, I mean, sort out all the global challenges that we face. Next slide, please. So, just as I told you that this nature is a gold mine of all the anti-inflammatory compounds. Now, you might be knowing that is right from 1897, the aspirin that was discovered from willow bark to paclitaxel that was discovered from Pacific View to artemisinin. Now, all these compounds were there. Now, did all these compounds have reached the market? Definitely, and we are using it in wide varieties. But yes, the bioactivities of these compounds, that is, whatever chemical compounds that have evolved over all these years, they have not reached market. Now, why has it not reached market? This is nature's pharmacy. Nature is actually storing lot many compounds that need to be taken into consideration. Now, how that can be? That can be definitely you have to use modern chemistry. Now, even though this nature was finding out, or the our nature has so many different compounds harboring in it, which is basically used for pathogen resistance and its surveillance in the extreme climatic conditions, our chemistry still finds, fails, I would say, or it finds we find it difficult to mimic those things. So it is very important to unravel the nature's pharmacy, because we don't need run around. We have to just think about ways by which we can in a very effective as well as safely we can use this treasure. And this treasure is definitely for mankind, which in turn will help us to find safer and sustainable ways for our living. Next slide, please. So now just go back. Yeah. So India's extraordinary biodiversity, that is what I would like to say, that we are one of the richest country in terms of biodiversity, right from Himalayas to coastal ecosystems to every Western Ghats. They all have untapped potential, right? Now this untapped potential has to be just discovered in a safer way, so that we don't, but we utilize them to develop novel drugs. Now, when I say the word novel, doesn't mean that something new. I would say that something that is potentially beneficial for us. It will be able to solve our health issues or whatever metabolic problems that a human system or the ecosystem or anything faces, that can be done in a very safer way. Now, in that means, then we have to think lot many things, that what are the limitations, what are the challenges, what scientific innovations we can put forward, so that our basic priority should be a global health. Until unless we are healthy, we won't be able to live at all. So just compare the cases of cancer or whatever metabolic disorder that is. I personally have one of my students that feels me very really bad. After second semester, she was there healthy, and finally she was about to complete her third semester. I came to that she has a metabolic disorder. And how I am helping her is I am keeping her online. I give her recorded session, because as a teacher I have a role in it. But I really wonder that she was totally fine. Now there was some metabolic disorder, and she was running across hospitals to find out that what actually the reason is. It has not been found. So don't you everyone feel that there is importance to find new drugs that will be able to find out the reasons that why is it happening? Now this case is going to increase. We are definitely, all people, or we as a social responsibility, we are telling everyone that we have to lead a healthy life. We have started cooking our foods on our own, right? We have put up all our habits and try to live a healthy life in all ways, whatever possible. So, hence, I would say that since we are having a very extraordinary biodiversity in our country itself, we have the responsibility to utilize it in the current way. Next slide, please. Now, now traditional drug discovery. Now, the traditional drug discovery was actually, you might be knowing Vedas, so like Ayurveda. Now, Ayurveda is rightly called as a goldmine of anti-inflammatory drugs. Now, yes, it has tremendous therapeutic potential. Ayurveda is actually a holistic wellness. It treats whatever disease right from the root cause. Now, what happens is, even though all these are there, how many of you or how many of us know that yes, this traditional knowledge has to be taken into consideration? Why I would like to put up that point is, now this traditional knowledge was generated on the basis of most extensive kind of research. Now there were no laboratories at that time. Just think how many years ago all these traditional methods of drug discovery were found out. There were no modern technologies. Right? So what is the need of the hour is actually utilizing these traditional discoveries. Now, how does modern discovery work? Now, they actually are using computational analysis. We are doing computational analysis and we are trying to scientifically validate. There was no validation for traditional discovery, but we have to scientifically validate them. Talking about any traditional medicine, you might not find a single compound working out. If you go into the scientific, you would find that it has n number of compounds working in it. Now, why don't we try and analyze how these compounds are actually carrying on, or what is the mechanism of action of all these compounds which are in that traditional medicine. So modern drug discovery is actually using all these technologies. They are trying to analyze the mechanism of action, which is very important. Now that will help us to develop new drugs. New drugs are new and safer drugs. Why? Because it is from traditional drugs. Now all these compounds, they can be monitored in a way that actually, say, suppose a particular drug is being utilized for a particular cancer. Now, particular cancers definitely may be due to, say, overactive of any gene or something, a protein or something. Okay, that is not working in the proper manner. So, by understanding the mechanism or scientifically validating it, you would be able to know that at what step actually the process is happening, and where can a particular molecule of that particular drug can work on it, can work on that pathway, and thereby prevent the disease further. Next, please. Next. So traditional approaches were, sometimes it was accidental, and there was broad screening of natural compounds, and definitely it was a laboratory intensive. So when I compare this traditional modern technologies, computational drug design, high throughput screening, genomics and protease integration, artificial intelligence, this all if integrated together, we will get drugs in a more easier way, in a more safer way. Now, I'm not talking about that you generate drugs, you sell it on market and become rich. I'm not talking about that. I'm rather talking about the social responsibility that we have. See, we have something, a treasure that we are not looking into. We have a treasure, we have a tradition, we have a knowledge system which has many drug molecules. Now, computational drug design will help you to curate drugs which will be able to enter the system easily, which will be more effective, which can be utilized by every person. Okay, now this high throughput screening, that is a modern methodology, it will help to screen molecules very effectively. And everyone says that artificial intelligence is going to replace humans. But I would say that artificial intelligence is rather going to help us it in a better way. Because if you try to incorporate this artificial intelligence, you might reach a result or the end point of your experiment in a better way, in a more effective way, and with less time and also cost effective. Okay, so that is what we lack. And for example, if I used AI or say several softwares available, you can actually see that, say, a particular molecule, say quercetin. You might find quercetin, or you might have read that quercetin acts as an anti-inflammatory compound for a particular target. So just put that quercetin into a particular software. It will help you to identify n number of targets that are present in your system. Now, those targets may be responsible or may act an important role for several diseases, for several health issues in our body. So what happens? It's like one molecule and you're targeting so many things. So that knowledge needs to be generated, because that will help us to discover a drug more effectively. So it is important to analyze what is the traditional system and how we can effectively implement these modern approaches. Next slide, please. Next slide. Tap it. Yeah. So now I was saying that it was like precision guided drug discovery. So when you think of precision guided, there are definitely certain challenges, such as molecular complexity. Now, which actually is the active compound, as I told you. This nature is actually juggling around so many compounds. You have to find out a particular molecule that is very effective. Okay. Now, the second problem that comes into being is the synergistic action. Now, what happens is in plant itself, you might have heard about that there are so many secondary metabolites. Now, you might have heard that a particular compound is being isolated and it's been utilized as drug. Now, a plant is providing a wide array of anti-inflammatory compounds. Now, if we use that plant as a whole or a part of that plant to develop a particular drug, now not only that particular molecule is acting, there might be so many molecules, so they might act in synergy. When they act in synergy, only then it can be able to produce a particular effect. Alone, it won't be able to. So that is a major challenge in natural product discovery. You have to think that how can that be utilized? We have to get that synergistic action of that particular compounds. So that is a problem. Second is sustainable sourcing. That is scaling products. That is, I mean, you are scaling up all the without depleting the wild population. Now, what happens is we as humans, when we find that something is beneficial, we just dig on it, right? We utilize in a way and we don't think of restoring it anyway. So sustainable, sourcing is very important. You are trying to utilize something, then you should put in methods also whereby you can actually maintain it so that it can be utilized in a proper manner. Going on to the technical limitations is if we have to use, now everyone cannot think of drug discovery, definitely we have to, because we require high level, mechanisms, or you require very important tools to analyze it. So that is also a modern challenge that we actually go through in natural drug discovery process. Because I myself had to, during my research work, I have to hunt around so many techniques to finally come and realize, okay, this technique is giving me a final result, which is able to justify what I am doing, right? So that is also a major challenge that we have facing in natural drug discovery process. Next slide, please. So as I told you, cutting edge methods, that is, we have to do a drug discovery in terms of highly curated manner, in a way that we can utilize all the methods such as high throughput screening, which is an automated, in which mainly automated systems are being used. Now, all these automated systems will be able to test or analyze the natural extracts, and it will be able to even replicate them in a manner that we cannot even think of. Now, genome mining is that is our genome. It is actually a storehouse of so many important things. We can actually activate the genome and develop certain. Now, this genome can be of a microorganism. It can be of a marine organism. All these are important sources. Not only plant world, even our soil. We can easily do the genome mining. So cutting edge methods, they are trying to transform this drug discovery. The only thing is we have to think on it. We have to think on these methods by which we can develop novel drugs. Novel drugs in terms that are, as I told you, about its efficacy and its safety. That is what matters. For you might be knowing there are so many drugs. We get to know a fine morning that this drug has been banned. It is no more in the market. Why is it so? Because it has been found that when a particular person has been affected with some kind of disease and all, and doctors, they diagnose it, they actually tell you that it was due to the dosage that was given to that person from long time. He might be treated with a particular drug for a long time, and is the side effect of that particular drug. So that is what I am trying to say, that we have to use cutting edge methods. We have to use procedures in which it will help us to transform this discovery. And definitely AI and machine learning, I told you, integrating genomics, proteomics, as well as metabolomics. This will help you to optimize whatever drug candidate you are trying to put up in the market. Now, integrate all this. Genome, you are going to control all the genes, all the proteins that are taking part, and of course the metabolic pathways. We have a control over it, right? Now, see, there is wide occurrence of even autoimmune disorders. You might be listening, right? So many autoimmune disorders are there. So we have to integrate AI and machine learning into it and utilize what is the positive factor. The only problem that we all think is we always think of the negatives. Definitely it has to be looked into, but why not utilize the positives of all the technologies that are being developed? How did AI develop? AI developed because of our own resources, right? Now it is the outcome that is, I think everyone who is sitting out here, they are using AI in some way or the other, right? Even though we are saying that it's going to replace human. So why not utilize for the betterment of human? That is what I am trying to put forward through my topic today. Next slide, please. Next slide. Okay. So artificial intelligence, as I told you, it's actually revolutionizing, right? I mean, the scientists are trying to use. Even we are thinking of next project of putting in AI-driven natural product discovery, because we have ample, results of in vitro experiments. We are doing in vivo experiments as well. So now we are trying to incorporate this AI technology, so that this technology will help it to analyze what actually the molecular structure is. How will it really interact? Now, if we interact that particular molecule with another molecule, what can happen, or will it be modulating a particular process? And definitely, my researchers in the lab, they are trying all many ways, and definitely to develop anti-infective compounds. For example, you might have heard of a trabectin. It's a compound that from marine source. Now that is an anti for rightly used for ovarian cancers. Now, since it was developed, now what other thing my lab is trying to might do, that I'm thinking of, is we might go into the drugs that have been already discovered. We will incorporate AI technology into it. We will incorporate machine learning into it. And then definitely I will be taking help from all those fields. So, and we are trying to find out that how this particular compound can be further modulated, can be further targeted. As I told you, that a particular compound it can target so many different proteins. Now, all these whatever resources that I have used till now, whatever softwares that I have, they've helped me in so many ways. In my research itself, I had identified 42 different compounds. Now, all the 42 different compounds, I subjected it to a particular software in which I could find that 42 different compounds were not only focusing the target that I was thinking of. There were so many other targets. N number of targets, and the 42 compounds that I had identified from a particular herbal decoction, it was used only for a disease, only for a particular disease or some certain symptoms. But I was really astonished to know that the compounds that are present in that particular herbal decoction could treat everything and anything that a human normally, or a human health normally gets affected with. So that is why I became more interested into drug discovery. I wanted to know that how actually this drug discovery can be utilized in a way that will help to identify newer compounds. Newer compounds, newer combinations, I would say. Now that won't reach definitely market in just a jiffy. It will take longer time. As you know, you might have heard that today all the drugs, with what ease we take in all the medicines. It has undergone lot many studies, lot many researchers, like 10 years of research and all. After having all that admit studies and all, because it's going to affect our vital systems. So it's not that very easy. But yes, I feel that I have a social responsibility. I have a social responsibility to identify molecules, give it to the market, so that the people who are more interested into it can actually work on it. We require basis. We require basis to develop drugs. You cannot simply put up a molecule into the market. Why actually that molecule is put forward? That is what we have to understand. Okay, so this is how I'm trying to do. We are trying to put in AI-driven natural product discovery, because that will ease the problem. That will sort out our findings. That will help us find newer ways. That will definitely help us to live healthy. Next slide, please. Now, this is another thing. Now, we have talked about sustainability. You all know it. Now, what happens is the Indian context is sustainable and ethical sourcing. Now, we have a rich biodiversity. Now, what I feel, maybe because I'm a proud Indian, I feel that India has the richest biodiversity. We are finding that whatever biodiversity we have, We have a role. Each one of us has a role to protect it. As I told you, once we find that this particular source or this particular plant, or let it be marine or microorganism, it is important in providing a particular, say, any anti-inflammatory, antibacterial, whatever antioxidant compounds we try utilizing. But we have important we should have an important thinking that it is a part of an ecosystem, right? This ecosystem needs to be maintained. What happens if you do a misbalance to this? It won't, I mean, you might think that it's not going to affect, it will affect us. Why? Because this ecosystem, if it is being imbalanced, what happens is slowly and steadily our biodiversity is going to go to a drastic stage. So maintaining the ecological balance is very important. Then only we have, if we are rich, if India is rich in biodiversity, I mean, remarkable biodiversity context, then it is also our responsibility to protect it. Now, traditional knowledge, as I told you, now you might find so many foreigners coming up to India, especially to Kerala. During their vacation time, we find so many foreigners coming up only for Ayurvedic chilsa. Chilsa means treatment. Now, why have they come over to India? Just think about it. Why have they? Because India is having that knowledge, the traditional knowledge system. Now, as Indians, we have this important responsibility to provide information, and we have to utilize the information correctly, and we have to put in our ethical standards. Right? Now, you might have heard about the traditional TKDL, that is traditional knowledge system library. They are trying to protect this indigenous knowledge. Why protect it? Because once it goes off, it is just gone. But since I am in Kerala, I don't have to hunt around for some immediate home remedies, because they are just outside my home. Just think of Tulsi. If I don't preserve that Tulsi plant correctly outside my home, what happens? The beneficial effect that particular plant is providing me, I would have to run around and find something a synthetic source. Why? I have something that is a nature's gift to me. My responsibility is to maintain it. So that is why government has led integration. Government is trying to provide sustainable sourcing. So this traditional knowledge digital library is actually providing protection to all the indigenous knowledge. Okay, this will help to develop more collaborations. Now, what as we people in this particular field have to think of is maintaining them. And these principles will definitely help us to develop drugs in a way that will be important for our welfare, and also maintaining the ecological balance, the ecosystem that we have to look into. Because you might be, if you have gone, you might have heard so many environmentologists, they all are working like anything. They are telling us that because of generation of this particular thing, or this has been introduced in the environment, it is going to affect, right? We are in our college, we are maintaining green campus. Now all this has become prime importance. So our role is to definitely utilize the natural source and also put in all our ethical considerations, and we have our responsibility to protect them side by side. Next, please. So now coming on to what happens in lab. Now you find some natural products. Now you have to develop them in medicines. So what actually is the primary or the most important step to develop a natural product into medicines is definitely the extraction procedure. Now you are trying to tap what is present in nature. As I told you, it can be marine source, it can be microbial source, it can be soil, it can be plant species. Now you want to tap something that they have hidden within them. or they're ready to give. The only thing is we have to choose the right protocol for extraction. Simplest example what I can give you is you take leaves of certain plants that has a traditional knowledge of being utilized to treat a particular disease. Okay, now Edathoda vasica, that is rightly used for cough and all. There are so many decoctions that are produced from it. Now you want to isolate or you want to utilize that particular plant leaves, you have to extract it properly. I would suggest that take two different samples. You take leaves of that plant. One set of sample of that particular plant can be shade dried. The other you put it in a hotter oven for you want it to be dried faster. Extract both simultaneously. Go for compound identification. Go for isolation. You will know the difference. Because the sample that was shade dried was allowed to dry in a natural way. Okay, so what happened through that is you will find mass difference in the isolation or the identification of compounds from the two samples. The thing that was shade dried showed that it had so many important anti-inflammatory compounds upon research, literature review search. And the other sample that was subjected to immediate drying through, say, oven or something, it didn't show the presence of that many compounds. So what happened? You have destroyed, by just choosing the long protocol, you have destroyed the secondary metabolites which can be useful for not many things. So just think that we people, we want to do everything in a faster way. We want to save our time. We want to get results, and the results should be that perfect. We don't want negative results. But why do we think that? Why are those negative results happening? Okay, so once you isolate the compound, you are going to characterize it. We characterize those compounds. We analyze those compounds in different methods. Then definitely the next preclinical and clinical phase is what we can do is we can approach pharmaceutical companies. They will do those studies. They will definitely do studies. And now, since they are integrating so many technologies, it can come up to the market in a more easier way. So what have we done? We have translated something, information that was present in the nature. We have translated them. We have translated them into medicine. So right from extraction, that if you take the source in the right way, even before extraction, there are, the secondary from time, like as the climatic changes, take a plant that has been subjected to drastic environmental conditions, take the same plant that is been growing in a healthy environment, you will get mass difference. What happens? Because you are providing, you are working in a kitchen that has no aeration system. You will be making a particular dish. You're working in another kitchen which has a proper aeration system. Doesn't make a whole difference. So likewise, we have to provide right from extraction or from the source. We have our responsibility to maintain it. We have to provide the ample ways by which we can actually utilize them, and we can definitely allow it to reach the clinic from lab. Okay, next please. So as I told you, this natural based drug development, that is right from extraction. Now, what traditional system has provided, we can rightly extract them. We can rightly extract them because, as I told you, you take a herbal decoction, you take a plant source, you take a marine source, you can extract in as many ways as you want. The only thing is, what are you looking for? You are looking for a particular compound, you can develop methods by which you can readily identify that. You can readily, yes, although there might be traditional knowledge, yes, that this is being utilized for a particular thing, but you have to develop a drug for clinical application, then you can definitely do in vitro studies. In vitro studies will help to scientifically validate. Now, once you scientifically validate, yes, say if you have a particular plant extract or whatever, the natural source extract, you actually put in the preliminary studies, you get to know that it's showing some result. Now, why is that result coming up? Why? You can definitely compare it to the already proven drugs in market, the synthetic drugs in market, compare to them. Already found out drugs, already developed drugs, you compare to them, and then you can see that, or you can rightly put up a chart in which you can say that this particular compounds that are isolated from a particular natural system stands out in which rank. Then you do all the molecular analysis, you do molecular diagnostic systems, you use molecular techniques. You can use RT-PCR, in which you can actually identify what happens to a particular gene in the presence of your compounds. You might be using as a single extract, a crude extract. Single extract, some people, they actually use the crude extract. Some people actually, they do extraction and develop different extracts that might be having compounds from non-polar to polar. So you can actually do a comparative analysis of all those extracts. So that will help you to identify or to know your sample or your natural source better. Now, what all this comes into being is you can actually successfully develop drugs that will actually able to cross that ADMET studies very effectively. Now, ADMET studies definitely, when we are doing so many, pharmaceutical drug discovery, ADMET studies plays an important role, right? Now, whatever you develop, it should be least toxic, right? Now, only then we can think of further clinical studies. You can send it for phase trials. Okay, so natural products can be rightly utilized only if we curate the pathway correctly. If the flow chart of how you're actually trying to develop a drug from a natural source is curated correctly. So you would have to think not only of a particular source, you have to also understand and identify where is that source. Are there any, there are environmental problems that it is facing? Because we have come across cases when I was doing in research that same plant, one of my co-researcher was working on a particular extract, the extract finished. So she had to again prepare the extract. So what we did is, what she got a result is, the result that she was getting for the earlier extract was quite different from the one that she was getting again, that she prepared secondly. So what we did is we used LCMS. You might have heard of LCMS. And we tried to analyze that how much is the efficiency in the concentration of the metabolites that are present in that particular extracts that was taken from two different plants, that is same, but it was taken from two different locations. So it was taken into consideration was what that environmental factors can affect. So right from source to the environmental factors to how we develop methods to analyze and further, justifying it with modern technologies and then developing it and bringing up to the market for further clinical trials would be the best way how we can utilize or skim out all these natural products. Next slide, please. So future perspective that I would like to say this, we know that our marine world is actually covering, that is, it actually covers 70% of the Earth's surface, right? It is still unexplored. So future of this drug discovery lies in marine world. The only thing is we have to optimize them. We have to optimize them side by side. I would definitely say we have to preserve them. Put in ways or put in strategies that should actually help to preserve them. We can rightly use our microbial world for drug discovery. It's beyond immense capabilities, but the utilizing it in the right way. So that is Then what happens if we utilize all this? And definitely nano carriers, very effective, right? Nanotechnology into drug discovery is now like we know from quite a long time, right? Now just synergistically putting in all these, the tradition and the technology, and definitely maintaining. So what will happen is synergistic action of all these will help us to provide better solutions, safer solutions, and sustainable solutions, right? Okay, next slide, please. So, concludingly, I would like to say that nature actually is providing unmet medicinal needs, right? Now, all these they are addressing. The only thing is we have to utilize it properly. Utilize it properly. We have this will allow us to go into a further important breakthrough, pharmaceutical breakthrough. We will have successful drug discoveries, which will be effective for our knowledge system, and definitely we would have sustainable practices. So that is what I want to tell each one of you. So the only thing is, if we utilize our nature correctly, this nature has immense number of prescriptions, immense number of prescriptions. Read it correctly, read it wisely, I would say. Wisely in terms that we can utilize the best part of it and side by side maintain it. Read through these prescriptions. If we read through these prescriptions correctly, we would be able to develop novel drugs, which are need of the now. Just think that so many drugs, there are so many side effects. Some are even depleting from the market due to some reason or the other. So it's high need to develop all these drugs. We cannot overlook this. We cannot. So when we have resources, the only thing is just look into it, read the nature's prescription correctly, and put in novel drugs. Okay. So next slide. So this is my lab, that is my college, and it's the information research lab team at my. He is my international scholar from Yemen, and one of them, that is the person next, second from the right side, she's actually a CSI fellowship holder, and others are doing their work in a very brilliant way. So we all are after drug discovery. We are trying to unravel several areas through this team, and we are trying to add on our social responsibility. We are taking into consideration. Rather, they are not. I've rightly told them that it is not you to get a PhD, but it is important that you have a social responsibility and have to look into it. Next slide. So this is my lab, in which we have a couple of projects. We have a patent. Now that was in association with Sujit. It was an Ayurvedic ear cleaning device. We got a design patent, and also we are working on all the phytochemistry. We have incorporated all the in silico strategies, computational, right from docking to MS simulations to everything. We are trying to incorporate it. And definitely we are thinking whatever results we have got is showing very good, and we are definitely thinking of putting forward greater solutions to drug discovery. Next slide. Thank you. That's it. Thank you for the opportunity that I've got through. I would like to know if anyone wants to know anything or would rather discuss or share your knowledge with us.
Yes, one second. Yes, we have questions. You can read from the question session, chat session. Or should I read?
Just a second. Just a second. I just want. Sujit, can I get this question? How do liposomal? Okay, yeah. Okay. Now I specifically address the pure aqueous soluble. Now there can be certain limitations. I think Pradipti, am I pronouncing your name correctly?
Yes, Pradeep.
Yeah. So actually, what happens is when all these nano carriers are used, there can be a problem. Is all the secondary metabolites, they behave very differently in different. Hello.
Yes,
Okay,
miss,
so what happens? Yeah, when you say about flavonoid and when you say of alkaloids, it's a huge group, right? Now that is a big group of secondary metabolites. So that might be the probable reason that in vitro release kinetics cannot be supported. Now thinking of a particular molecule, then it might help you, right? Now this nano carrier specifically, it can address pure acute solubility of flavonoids, and that is the complexity of that particular flavonoid or alkaloid group might be the probable reason. So, and in vitro release kinetics data support the superiority. Yes, definitely. Their release kinetics can be because of the huge group of molecules. Rather, if you isolate a particular flavonoid, you compare it, then definitely it will be it might give you a better result. What were the we are not actually into AI tools right now. We are, as I told you, my work will be definitely integrating. Right now, we're using several in silico strategies, in which right from talking to simulation studies, we are trying to incorporate how actually these molecules are integrating or reacting with the targets that we have chosen. And we are also utilizing several AI-driven machine. Yeah, AI tools will help in further. So we are actually into it. My lab is presently doing into in silico studies, which is providing ample information regarding how it is binding to the active site or not. We are comparing with the positive molecules or the already known drugs that are present in market. And further, we definitely will use AI tools, and we are in search for some technologies also to further develop the drug molecules. Now, all these machine learning models, whatever I have gone through review of literature and all, they are actually will help us to identify those genes that can be amplified. Amplified in terms that these gene clusters can be utilized for developing a particular compound, and that can be further been led into. Now, what we normally do is we don't simply take up all these softwares. We actually, I have always in my lab, we have taken into consideration that we do in vitro studies, we do in vivo studies, because all the technology that you take different softwares, we have got to know that the software that is used in one system, the result that we get from one system is a little bit different from the same result that we get from other system. So screening all those natural products, not only depending, fully depending on AI is not a very good thing. Rather, what we do is we predict these molecules after doing all the in vitro studies and in vivo studies. So that is how I feel that AI machine learning will help us to identify which particular gene mining or what particular gene mining can be done. Yeah, how do you say that? That is a very relevant question. That is what I was trying to emphasize. That is, how do you select a plant? See, selecting a plant is in terms that you have to identify what is the potential of that particular plant. Okay, you cannot simply the one of the major problem in natural drug discovery is purifying compounds. You require so much amount of money. You require it is not a cost effective procedure. So you would have to select plants in terms that are not explored properly, or even though if it is been explored, it is not explored in a way, say you don't know how actually the mechanism of action it is. So for screening, you have to check in plants which are, and you have to go for review of literature for past 5 years. Whenever you do a selector plan to selector, you should identify the gap area that is present in it. How can that be? Whatever studies are being conducted in it till now, you do a thorough review of literature, and then you will be able to identify what particular or which particular plant has to be known. Now you want to focus on a particular compound, then you have to identify the quercetin is not present only in one plant. That might be present in n number of. Then you would have to identify. You're working on quercetin, you're working on butylene, you're working on some other compound. You have to identify them, that where is this natural source present. Now you can compare them. So for screening, or you want to choose a particular, select a particular compound or a natural, only review of literature will help you. Find out, do a maximum review for last 5 years, what was discovered in that particular plant or the natural product. Then try to identify what is the gap area, and definitely you can select. Okay, so when you talk about Indian medicine, such as all the Ayurveda, it's a traditional medicinal system. Now, definitely in consideration with a very good practitioner, what I did was I had gone through almost something that was, I was focusing on all the medicines, traditional medicines that were used against the chronic inflammatory disorders. Now, when I looked into chronic, there was an endless list. Now, how can I find out that which is my requirement? So I approached an Ayurvedic practitioner who has actually immense knowledge about Ayurveda. Now, what that helped me is that person was able to tell me that this particular medicine is being prepared in a way that is going to focus on a particular area of that particular health. So reaching onto them will help to definitely find out that we can actually screen. We can think of plan selection, and also say any drug, any made up drug that is present in the market. It can be a combination. So getting in touch, this is how we have tried to integrate our knowledge. Our society is it is full of knowledge. You have Ayurvedic practitioners, you have pharmaceutical people, you have so many scientists, you have people working in AI, you have people working so many different biological technologies. We have to integrate all them. We have to integrate them and then utilize this knowledge for proper drug discovery. I hope I am answering questions, because I think there are people from many different fields and they are going to qualifications. Yeah, qualifications. Okay. Now see, what we got is what we should know, that you should have a basic knowledge. Say, what actually a plant is. I always tell my students that if you don't know about something today, it should not remain as unknown tomorrow also. Just Google it and find out. Now, for what qualification you have, you should know what is the basic knowledge of a particular field that you're choosing. See, but I'm not talking about the degrees, not MSc, not BSc. It's not about graduation or doctoral or postdoctoral. It's about the knowledge that you're trying to analyze. You have to understand what actually the basis of it is. Phytochemistry is a big area. I have just got to know a drop of it, because there are immense possibilities in it. So first one thing is, you have to put in all the resources. We have to utilize it. We have to try out ways by which we can develop our knowledge in all terms. Yeah, this is a very important additional pharmacy. Yeah, additional skills in terms is, you should be knowing about what are the in vitro experiments, right? Because test tube experiments are the ones that are going to prove what you are thinking of. In vitro experiments will definitely simple. You might have done qualitative or quantitative analysis. That should be first skilled properly. That should be so very perfect, because that is the basis of what you're trying to do. You might be trying simple chromatographic techniques. You might be trying HPTLC. That should be done in the precise way, in the perfect way. Because until unless that is not correct, that will not help you to find out further. Okay, so as pharmacy students, you should, whatever experiments, right from the basis, whatever experiments you are doing, get skilled into it. Later we can think of big, such as cell culture studies or animal studies. That comes later. Until unless your preliminary analysis is not correct, it won't help you to lead or put up any data that can be useful for drug discovery. Anything else?
Any more questions?
Now, someone asked about cost. See, right now I am utilizing my college, and during my research, I could get up a scholarship. So that is how I was able to put up whatever things I have acquired till now. So cost is, again, what we can do is, if you want to start set up a lab that will help you to develop a drug discovery, that definitely there are many government opportunities. The only thing is, you have to find out people who are good enough in that particular field, because a person without knowledge will not be helped by anyone. So you would have to find out people that will help. They will have immense information, or you can say right information, so that in that way definitely we can put up projects into so many resources of the government. Even some private sectors also, they are providing fundings. The only thing is, we have to show them that we are capable enough to do it.
Okay. So I guess we have finished on time, 4 o'clock. So yeah, as far as the certificate question is there, can you mute too? It's like going.
Yeah, sorry. Yeah.
Okay, so as far as the certificate is concerned, those who have attended can message me your name and your Zoho ID. We will issue the certificates. And we already mentioned that those who are present for the full session gets the certificate. That's the only requirement that we place before the candidates. So I guess there are no more questions. So I will take this opportunity to thank Dr. Tina ma'am for accepting our invitation and delivering a thought-provoking and actually a very insightful session. I'm saying this because you mentioned two things which are actually considered contradictory. One is preserving nature, because that's one thing which comes under environmental responsibility. But if you consider scientific discoveries, then that it is considered contradictory. But in India, as you rightly said, we can manage that because you have to take up that only if there is nature, then there can be medicines. That's the main. I guess that's something which we take away from this particular session. I guess you have already cleared very many basic concepts with respect to drug discovery from nature itself, and there was heavy focus on sustainable research. And you also brought up AI into it because before going into AI, it is very much important that we understand our nature and the medicine or the medicines that can be obtained from them. I guess the questions itself shows that the participants were fully involved and they were very enthusiastic. And I'm really thankful to the audience also for keeping this session alive. And once again, thank you, ma'am, for attending this session or taking this session. And as far as the audience is concerned, it is because of them that we are able to continue with this session. So they are also contributing to this Saturday Knowledge Series. And I guess every session we are moving in the right direction of ensuring that we have a good, knowledgeable, quality scientific manpower in our country, which many of the government projects also, they are also in line with this vision. Many of these colleges also have their vision with respect, which is in line with these concepts. So thank you, each one of you. Anything to add, ma'am?
No, thank you, Sujit. Thank you, Spectratix. Thank you, audience. I think there are a lot many learned people in this. Anything, wrong that has been said from my side can be, I think, taken care of.
You also gave me an
So,
opportunity to sit in the classroom once again.
and it was good for me to, speak to unknown people now who are really interested in this field. Let it be
Main
10
thing
or
is
few, it's fine enough. But what I am trying to say is that our social response is one thing that needs to be taken care of. Now, we all can bridge together. We all can bridge together, because see what knowledge I have, that is how we came in touch, right? Your knowledge and my knowledge can be the right way. There are so many people who are ready for sustainable developments. They can be incorporated. Definitely we can develop. We can develop in a way that will help us in all means, and likewise unknowingly we'll be doing our social responsibility. That is very important, and we can pass this information to our generations to come.
There's something,
that will be good.
as you rightly said, there are many students, there are almost 200, 300 people in this group, and 30 to 40 they join depending on their expertise. So there are many people who take advantage in their PhDs, research activities. So that's something, means it's sort of a group being formed, so where we help each other in science, and anything related to chemistry, anyone can also. So we'll break for the evening. Any more questions? Okay, ma'am. Thank you so much. See you soon.
Thank you once again.
Okay, bye everyone.